Welcome everybody to our BAGIM seminar for today, the 19th of October. So welcome to everybody. I'll give you a few little rundowns. I'm trying to admit people as we go. So if I'm looking to the right, that's me letting more people into the meeting. All right, so we'd like to first of all thank all of our sponsors. Oh, the sponsors have gone away. My keyboard has decided that it's taking control of my life. It is just doing whatever it wants, apparently. Let's see if I can, we're just going to do this manually. Okay, so yeah, thank you to our sponsors. We have Schrodinger, OpenEye, Chemaxon, Cresset, all of our wonderful sponsors. We thank you all so much for supporting us. We are definitely a small community, largely just focused on trying to, share good science with modelers of all varieties all over the area. Currently, looks like we're hitting Boston and the most of the west coast of Europe. So that's great. If any of you would like to become a BAGIM sponsor, we are more than happy to take your money and your support, but you get something out of it as well. So we have the silver, gold, and platinum levels. You can either get recognition at meetings and online in BAGIM. At the gold level, you can chair a session. So that means I don't have to do anything, which is great for me. At the platinum level, you get to sponsor your own speaker, which is what we have today, which we are very, very happy about. This is who we are. This is the current BAGIM Executive Committee. There's Brian Kelly, Tony Arvanites, Scott Parker, and myself. And we don't have that many job postings. That is one of the things that we do here is if people have jobs that they like to share, but Scott just shared with me the job posting that we currently have available at OpenEye. So if anybody's interested in those, go ahead and either ping Scott, head up their website, take a look, see what's going on. And so for today, our first speaker is Julien Michel. He is a fellow at the University of Edinburgh, and he will be talking about the energetics of protein disorder order transition in small molecule binding, followed by Jenny Brookes, who is an application scientist at Cresset, and she will be talking about free energy perturbation to prioritize the best molecules in early drug discovery. So just a few ground rules. If you would like to ask a question to either of our wonderful speakers, totally fine. Put it in the chat, and at the end of each speaker's section, I will call out the questions in the chat and ask you to unmute so that you can ask your question yourself. This sort of controls for a little bit of the wildness of Zoom and helps to minimize any interruptions. So I'm going to stop sharing now, and I'm going to ask Julien Michel to please share your screen for me.
Sure, no problem, Rebecca. Let me get started. All right. And I think that's me. Good to go?
Looks great.
All right, okay. Well, so thank you for the invitation and the opportunity to speak at BAGIM today. It's exciting to be in touch with colleagues in the US. I haven't gotten the opportunity to come over there for some time, as you can imagine. So I'll be talking today about work we've been doing for a number of years, actually, but we're only starting to publish it. And so we're going to have a discussion about protein disorder, structural disorder, and how this may turn out to be something that could be exploited for small molecule drug design. So just to set the scene, so I'm sure many of you are well familiar with the concept of intrinsically disordered protein or intrinsically disordered regions in protein. And essentially what it means is that change in disorder in protein structure are used a lot by nature to drive formation of, for instance, protein interactions or protein DNA interactions, or generally to regulate physiological processes. And I've shown a few examples of what that can mean in nature and with biomolecular structure. So you can see, for instance, metal templating folding of a protein, or you have trimerization of disordered monomer forming a partially structured trimer. You can have ordering on binding DNA and also ordering on binding another protein partner. So these recognition mechanisms are very important in nature. And essentially the question we seek to answer in this research here is, can we exploit similar type of disorder order transition for the design of small molecule ligands? So in this work, we've chosen a model system to study this problem, and that's the protein MDM2. So MDM2 is a fairly well-known cancer target, and it's been the target of a number of drug design program by many companies over the last 20 years. And for the purpose of this talk, what we're interested in is that the N-terminal domain of MDM2, depicted here, contain a so-called lead region here, which are the first 24 amino acids of the sequence of the N-terminal domain. And that region is known to be unstructured in native condition and to exchange between unstructured open or closed states on millisecond time scale in vitro experiment. So the main binding site of the N-terminal domain of MDM2 is here, and it normally binds to the tumor suppressor p53. So the strategy to block the activity of MDM2 is to develop an antagonist of this PPI by occupying the space here. So you can see it's in the vicinity of this intrinsically disordered region. So initially, not much was known about this idea because it tended to be even truncated in construct use to study protein-ligand interaction on this system because it made the biochemistry a bit more tricky. And there was also no evidence that it was particularly important to begin with. So some of the earlier antagonists, for instance, the nuclein compound developed by Roche, there was no evidence that there was lead structuring or interaction of the nuclein with the leads on binding. Indeed, we did some work a few years ago where we showed by MD simulation that the lead appeared to remain closed and disordered. But if you contrast here that scaffold, piperidinone family developed by Amgen, this was reported to have a very interesting effect on the lead preferences. So we can see here by X-ray, NMR, and other sources of evidence that the lead orders on binding this particular scaffold inform this helix turn extended motif here. So we wanted to investigate what is special about this scaffold that makes the lead ordering feasible and not that one. So one thing we had to do to do this work is to generate the data that would allow us to rigorously compare binding affinities in the absence and presence of the lead region for these two scaffolds. And we had to effectively make our own measurements here because the data was incomplete in the public domain and also was done inconsistently with different construct or different experimental protocol that turn out that they do matter and influence the binding affinity measurement. So to really have a good handle on that, we use a consistent protocol where we minimize the effect of affinity tag, for instance, and also the effects like DMSO to get hopefully high quality data. So we can really look for binding selectivity trend in the measurements we're doing. So having done these measurements, we were able to measure the binding affinity of nuclein 3A to a construct of MDM2 that include most of the lead, at least enough residues that we don't think that what's missing could be involved in interactions. And then a very truncated version where the lead is so short that it cannot really undergo structuring on presence of a compound anymore. So what we've seen with that is that there's relatively little effect in our say entalpogram here in the ITC measurements for Nutlin 3A in the presence of absence of the lead region. But in the case of AM7209, there is a very significant effect. So MM7209 is extremely potent picomolar binder. So we need competitive titration protocol to measure its affinity. But in the context of the truncated lead, it's a much weaker binder, similar in potency to Nutlin 3A. So this is just looking at the previous data, but summarized in bar plot, where we can look at change in the thermodynamic signature of binding between these two compounds. So if you look at Nutlin 3A, you can see that the enthalpy of binding or the entropy of binding don't really change, but when it's binding to the lead or the lead short variant of MDM2. In the case of AM7209, we see that if we truncate the lead, we lose significantly in enthalpy of binding and we gain significantly in entropy of binding. So that's telling us that there is fundamentally something very different going on with the lead in the presence of this compound. And we see that there is overall a 250-fold preference for AM7209 to interact with constructs that include the lead IDR. So it shows that this unstructured motif is actually very important for recognition. So the question then is, how can we use a computational approach to help interpret these measurements? And here we've combined two approaches. In the first one, what we've done is we mapped out the free energy surface of ApoMDM2. And to do that, this is not using a methodology that we developed. Essentially, what we've done is we combine multiple different techniques developed by other groups to get to that sort of free energy map that help us understand the conformational preferences of the lead. So to briefly summarize the approach, initially, we use the accelerated MD method, originally developed by the McCammon group, to sample lead conformation in ApoMDM2 using different starting states of the lead. So some that look closed, ordered, or disordered. We then pull the data we get from this simulation and we find a 2D projection along two collective variables that can separate open and closed lead states so that they appear in different parts of this map. Once we have a projection that allows us to resolve the states of interest, we carry out umbrella sampling simulation using AMD snapshot as starting point. And once we've done this, we reconstruct finally the free energy surface. So we know about relative population preferences in this projection using the variational free energy profile methods developed by the Darden lab. So what do we learn then, having applied this methodology? This is our free energy surface of ApoMDM2, including the lead IDR. And we've categorized that into different regions. So you can see here in that part one, we see what we call open and disordered state. So there is access to the p53 binding site, but the lead is unstructured. Here, that's our major state. We see primarily what we would call closed disordered state. So the lead is closed. There's no access to the p53 binding site, but it's remain unstructured. And here we find 3, 4, 5, we found different flavor of order in the lead that leave access to the p53 binding site. Now, if we integrate the free energy surface and look at relative population, we find that these closed disordered states account for about 80% of the population of the lead. And this ordered state accounts for 6 to 8%. And so overall, that means this open ordered state is less stable than closed disordered by approximately 1.5 kcal per mole. Right, having established that, we want next to look at the free energy of binding of Nutlin 3A and AM7209 to these different lead states that we've identified in ApoMDM2. And here the methodology that we use is an alchemical approach, so an FEP approach, if you're more familiar with that acronym. And what we're doing here, though, is that we're carrying out absolute binding free energy calculations. So where we take a molecule of interest, and essentially through a thermodynamic cycle, we remove it completely from the protein ligand complex or solution. And that allows us to measure directly, to compute directly a standard free energy of binding that we can most directly relate to our ITC measurements. So what we've done here, though, is we've developed a variant of ABFE calculation, which we call adaptive ABFE, which is done to optimize resources allocation to decrease computing cost. And the reason we have to do that is that we're pushing hard the methodology here. It's usually not applied that routinely to complex molecules like AM729 and proteins that have flexible parts like the disordered lead region. So I'm going to give you a little bit more detail about why and how this adaptive ABFE protocol work. So the first thing to realize is when we carry out an ABFE calculation, we have to define a so-called lambda schedule where we start from an interacting state, say a MDM2 ligand, and we go through a schedule where we gradually decouple that ligand from the environment. So we can turn off electrostatic interaction and then turn off Lennard-Jones interactions typically. And that has to be done in the complex and also in solvent. And when all of that is done, you get your free energy of binding by some post-processing of all these independent simulations. So that's what I call a static alchemical workflow where we set the lambda schedule, we assign a fixed amount of sampling to each windows, and off we go, and we run also calculation. When they're all done, we analyze them. So we've done that here using as a model initially PIP2, which is a smaller, simplified variant of AM729, because we wanted to have a go at this using more tractable compound to begin with. And we looked at MDM2 with a truncated lead variant where we have to worry less about sampling to begin with. So you can see here that's 5 replicates, data from 5 replicates of this static protocol. And we're looking at how the free energy of binding of our compound converge as a function of the total cumulative sampling time used across this lambda schedule. Well, you can see that eventually, if you put enough resources, you start to see mean free energy estimates that's relatively stable. But you can still have significant scatter between different replicates up to 2 kcal per mole. So that's why you need about 5 replicates here to have some confidence in your free energy estimate. And so if you're pushing that far here, you've used about 20 microsecond of cumulative sampling time. So that's a lot more than what you would do for relative binding free energy protocol for sure. Now, if you look more closely at the lambda schedule, though, it turns out that the rate at which the different parts of the calculation converge is very uneven. So we can see that the vast majority actually is fairly well behaved, and we don't need to run for that long to get very stable estimate of the free energy change. So we're looking on this plot here at the standard deviation between our 5 independent replicates of the uncertainty of each of the 70 or so window in our lambda schedule. So what you can see that there's only a few here windows that contribute most of the uncertainty to the overall binding free energy. And so that is the reason why using an adaptive sampling protocol here can be quite cost effective. So what we do here instead is we carry out our setup as done previously, but we only carry out MD simulation for a short amount of time initially. And we pause and we look at the rate of convergence across replicates of our different simulations. And on that basis, decide which simulation needs to be extended by another chunk of time. And by doing so, eventually we terminate early part of the schedules that are well behaved, and we only allocate resources to the parts that do require longer simulation because the free energy estimate is noisier. So if you do that, and you repeat the calculation on the same system, you can see with the adaptive protocol So the dashed line here are epochs. So that's when we pause a calculation, analyze the data, decide how to allocate resources and carry on. But when we carry on, we use much fewer resources because many windows have already converged, that is after one iteration. And so we find that eventually we get to virtually the same answer as using the static protocol, but on this particular example with about 6-fold less compute resources. So of course, we need some threshold parameter to decide whether to stop or carry a simulation further. So that needs to be investigated. So if you set this threshold parameter too low, then you don't have much savings overall because you keep progressing all your simulations. Eventually, you still end up with essentially the same answer as before within your confidence interval. If you set it too high, so then you can have premature termination of your calculation, and you end up apparently saving more compute, but actually your result is biased and is significantly different from the brute force answer. For that system, we find a sweet spot of about 0.1 kcal per mole, although that probably needs more research to know if how well it generalizes. So having done this, having validated the adaptive ABFE protocol on PIP2 for lead short, we then extended it to AM729 and Hotlin 3A, looking at 3 different states of MDM2. So either lead short or 2 variants coming from the free energy surface of ApoMDM2 with a full lead, where the lead is either closed or ordered. And what happens there? Here are standard free energy of binding once we finish running the adaptive protocols in every case. What have we learned here? So looking at Hotlin 3A to begin with, we see that a small improvement in free energy of binding to lead close or lead order with respect to lead short. We find that there is preference for stabilizing the ordered state versus the closed state by about half a kcal per mole. If we look at AM729, we see a fairly different trend. We see that binding to lead close is actually disfavored compared to binding to truncated lead variant. And binding to the ordered state is much more stable versus the lead closed state by about 3 kcal per mole. So if we take these results and we combine them with what we've learned from the free energy surface of ApoMDM2, now we can explain what's happening and why we see a difference between these 2 ligands. So in the case of ApoMDM2, the open ordered state is about 1.5 kcal per mole less stable than the closed disordered state. Now in the case of the Hotlin 3 MDM2 lead complex, open ordered state is preferentially stabilized by Hotlin 3A by about half a kcal per mole, but that's not enough to overcome the intrinsic instability that's seen in the Apo free energy surface. And so we still mainly form a closed disordered state in the complex. In the case of AM7209, the picture is different. Now the selectivity of AM7209 for open order is sufficient that it overcomes the instability that we see in the Apo free energy surface. And so we end up forming the open order state mainly in the complex. Now we can do more than explain qualitatively what happened and why there's a difference between these 2 ligands. Looking back, we can see that we can use the ABFE calculation as a technique to find compound that can be designed to order the lead if that's the design goal. And so here there's a simple criteria: maximize the gap in free energy of binding between lead close and lead order states, and that should help form mainly these states. We can also look more closely at states of the lead to try to understand why there is a difference in binding between these 2 compounds and the lead. And of interest is the fact that lead closed state is much less preferred in the case of AM729 versus Hotlin 3A. And what we look closely, we see that although we call these states lead closed, they do differ. If you look more closely, you see that AM7209 has a bulky substituent on its piperidinone scaffold that sits above helix alpha 2 here, and as a result, it's pushing away the lead, which doesn't happen in the case of Nuclein 3A. Now, in ApoMDM2, when the lead is closed, it also likes to sit above helix alpha 2. So that gives us the hypothesis here that we can destabilize the lead closed state by introducing steric bulk to prevent interaction of the lead IDR with helix alpha 2. Okay, so with the time that's left, I'd like now to ask a related question. So hopefully we've come up with explanation why we see differences between the binding of AM729 and Nuclein 3A to MDM2. But what we thought was needed attention is to also establish why does the lead ordered state like to form. It's not stable in ApoMDM2. So what are the thermodynamic driving forces for adopting this state in the presence of AM7209? So to do that, we decided to focus on the lead ordered state seen in our simulation and initially explore computationally what we can do to destabilize that state by introducing mutation all along the lead IDR. And then on the basis of what we were seeing in our simulation, select mutants that we could prepare to characterize them by ITC and NMR. And so starting therefore from equilibrium simulation of AM729 in complex with different MDM2 mutant, we looked at a number of structural parameters, and one that turned out to be quite useful is to monitor the helical propensity of the lead region. You can see there's this helix, short helix at the base of the lead that is only seen in the ordered state. So that's how it's about 30 to 40% of the time present in our wild type simulation. And you can see that when we introduce some mutation, for instance, in that part of the lead, they don't necessarily have much of an effect. And as we get a bit closer to the base of the lead, some of the mutation start to significantly change the helicity of the lead. One that caught our attention was I19G, so isoleucine 19 here, mutated into glycine. You can see it almost completely destroys the helix. And this was clearly related to hydrophobicity of the mutant. You see either I19A is somewhere intermediate, and I19V is similar to wild type. So we thought that was quite an interesting observation. So overall, we picked a number of mutants based on this scanning of stability of the helix. And then we carried out ITC experiment on this mutant. So what you can see first, Nuclein is a negative control. It essentially shows that no matter what mutation you introduce, you don't really see much of a change in the affinity of Nuclein 3A. So that confirmed that the lead doesn't really seem to play an important role. In the case of AM7209, remarkably, we see that mutation can modulate the affinity of AM729 up by 3 orders of magnitude. So I19G here, for instance, makes AM729 a 4 nanomolar binder, whereas in wild type it's 4 picomolar. And that's also, we can see there's something going on in the thermodynamic signature of binding. If you compare AM7209 thermodynamic signature to the lead, you see we've got this unfavorable entropic contribution and a large enthalpy gain. If you look at the truncated lead, we see we've got a more favorable entropy of binding. And if we take I19G, we see that qualitatively the signature looks more like that of the truncated lead. So for us, that's evidence that we are interfering with conformational change of the lead on binding. So we picked that mutant to investigate this in greater details using high-field NMR experiment. And so if you, broadly speaking, with this experiment, we're using label samples of MDM2 wild type and I90G mutant, and we track chemical shift of the backbone amide group. And use those to infer whether we see change in structure or dynamics of MDM2 on binding of AM7209. What you're looking at here on this graph is a result of the so-called chemical shift perturbation analysis, where we're looking at how much the chemical shift of nuclei in each residue are changing on binding of the ligand. So if we see large changes, that's evidence that the local chemical environment is changing, either because the ligand is now interacting with this residue or because there's been a conformational change in the protein.
Julien, we've got to give you a 2-minute warning. We're a little bit over time already.
Sure, yeah, no problem. I'll wrap up in within 2 minutes then. Okay. Okay, so you can see in the case of wild type, we see there's a large chemical shift CSP values in the lead region. And these are almost completely gone in the case of I90G. So that's strong evidence that there's differences in interaction or conformational change between the mutant and wild type. And we can also show that using a nuclear spin relaxation experiment, which give us measure of change in fast dynamics of the MDM2 protein. What this plot tells us is in ApoMDM2, this low NOE value in the lead region shows that this part of the protein is unstructured. They tend to increase and become more similar to the rest of MDM2 in the M7209 complex, indicating of ordering. Whereas in the case of I90G, we don't see this qualitatively a significant change in NOE profile. So further evidence that there hasn't been structuring. And so we can then put all of this together and reconcile this observation with additional free energy surface calculation. So using the same approach that I've outlined before on ApoMDM2, but this time carried out in the complex with M7209 of wild type or mutant. And then from the lead property of the free energy surface, calculate, for instance, the overall secondary structure preferences. We can see that there's low helix population in M7209, in wild type, sorry. But in the M7209 complex, which is this increase in helicity, which is, we only see a small increase in the I90G mutant. And then finally, we also did the experiment, simulation, where we removed this chlorine and replaced it by hydrogen atom in M7209, and repeated this calculation and seen that we ended up with a profile qualitatively similar to I90G. And so we use this to conclude that what is stabilizing the order lead state is what we call hydrophobic collapse of the lead motif. So we see that this pattern of chlorophenyl substituent, then the threonine 16 here and I19 that cluster together in the ordered state. And if we introduce change in either of these residue, we see significant destabilization of the lead. All right, so I'm just gonna wrap up here. Hopefully I've shown that it could be beneficial to think about how to induce ordering in IDR with small molecule. We can think about using techniques like free energy surface calculation and binding free energy calculation to design this compound or to generate hypotheses. In the case of MDM2, we think that ordering of the lead IDR is driven by hydrophobicity of the resulting complex, and we hope that this observation may help identify other IDRs that could be ordered by small molecule in other proteins. And with that, I will stop here, and if there's time for questions, I'll happily answer a few.
I think due to time constraints, I think if anybody has questions, please put them in the chat. Julien, please feel free to reply in the chat. If we have time at the end, we'll do questions then. Jennifer, would you be willing to share your screen real quick?
Yeah, I'm sharing now, I believe. Is that the case? Can you?
Try one more time.
I can try again.
Wonderful. Got it. Looks good.
That's working now. Great. And I take it everyone can hear me okay?
Can hear you just fine. Go ahead.
Great. Okay, so I'll get going. So thank you for having me here. I'm very excited to be talking. And I will just dive straight in. Okay, so many of us probably know that the typical drug discovery workflow can be leaky, inefficient, costly, and you can actually either waste time on bad molecules or possibly even miss good molecules. So the workflow that I'm going to show in this presentation will simulate a drug discovery pathway which is quick and successful, starting with casting the net wide at around 100,000 fragments explored. And this will take just a few minutes. To then filtering 400 of those compounds, which can take actually just a day and a half, then filtering 40 of those in just a day, and then refining 20 promising candidate compounds in just under half a day. So all in all, the hope is to demonstrate that it is actually possible to discover a good high affinity binding drug all within the space of a week. So to do this, we're going to start with using Spark, which is a bioisoster replacement solution. So starting with a molecule that shows interesting properties even. For example, good biological activity. Spark will suggest biologically relevant replacements, or in other words, bioisosters for key fragments. The fragments found by Spark will mimic the 3D properties of the original fragment in electrostatics and in shape. And this approach can be used in a variety of the experiments that are listed here. But for this case, we're just going to focus on simple R group replacements. We'll be looking to find alternative binders with potentially improved binding affinity. So what happens in Spark is you select a region to replace and remove the atoms. And the geometric distance and angles between any of these broken bonds are recorded. Spark then searches a database for any fragment that has the right number of connection points that geometrically match the broken bonds on the target. And these matches are considered on the basis of both angle and distance. From this, products are formed with these fragments that have the correct angles and distances, and the connection points are merged, and you get what we call the product. This product is then minimized, and we add what we call field points. And then from this product-centric view, we're generating a really wide range of biologically relevant bioisotopes. Only now, when you've got the full product molecule made from these fragments, is the bioisosteric replacement then scored against the starting structure for electrostatic and shape similarity. So that particular example was actually for scaffold hopping. I just want to emphasize for an R group experiment, there's actually an extra step. So as there's only one actual attachment point in an R group experiment, Spark actually generates a set of possible result molecules, roughly 20 or 30, by actually rotating around the fragment around the attachment point. So the rotamers are explored, and the rotamers with the highest similarity to the reference is the one that's actually kept. So that is the Spark contribution. After this first Spark phase, we will then use Flare FEP, or in other words, free energy perturbation in our workflow. So Flare FEP is based on exciting collaboration with Julien Michel, who you just saw gave a fantastic talk. And it builds upon the top of the best open source tools and combines them into the Flare interface you see on the right here, using a fully automated workflow that has been internally validated using different data sets. So Flare FEP in particular uses a significantly improved and extended version of LOMAP here. And the user gets full control of the simulation parameters both within Flare, and you can also use the Flare Python API. And it's essentially used to calculate the binding affinity to small molecules. And as for an example, please see this very recent publication that I referred to. So FEP is particularly useful for a typical drug discovery problem when we have a ligand A that has a known activity and we have known ligand protein interactions, possibly through X-ray data. And what we want to do is start designing congeneric ligands such as ligand B down here. So given that we know the activity of A and the interactions that it makes with its target, can we do this? Can we predict the activity of B? We might not have any data much for B, not even any X-ray data, but we do need to be confident that it shares a common binding mode with ligand A. So we can actually do this. We can calculate binding affinities by exploiting this thermodynamic cycle. And I just want to emphasize that taking the horizontal routes is not really computationally tractable. So that would be having these ligands go from the solution state to binding to the protein complex. And that just takes an awful lot of time, there's a lot of noise, and it's hard to find convergence. If we're to go down the vertical paths in the thermodynamic cycle, then actually we can much more easily find the delta G between A and B by calculating the transformation from A to B in the solution phase and also in the protein complex phase. And this will give us the difference in binding free energy between A and B. So what we do in this type of calculation is ligand A is literally gradually morphed into ligand B via an alchemical, or in other words, non-physical intermediate state. And we do this via what we call a switching function, a switching lambda function. We can also, in these transformations, include intermediate states, which can facilitate the morphing from ligand to A and B, but it depends on the complexity of the transformation. So a bit more on that later. So critical to the success of an FEP calculation is actually setting up a perturbation network that links up all of the ligands and their transformations. So in Flare FEP, we actually use two types. So one being a fully multi-connected network with many links versus a star graph, which is actually one link per compound, or perhaps more if intermediates are included. So the star graph is actually a lot faster to run. So we will start with a star graph initially. And it's particularly useful for lead optimization, excuse me, where you're modifying an R group of a certain molecule for improvement or variety, which is exactly what we want to be doing here. So in both, you need at least one binder for a production run, because you need this in order to start to make binding affinity predictions. And you can also choose between using single versus dual way calculations, which in other words means making the transformation from ligand A to B and then back from B to A, so both forwards and the backwards direction. Of course, this means that a single way will actually take half as long as run than a double way, a dual way. So a single way can be incredibly fast and very useful for lead optimization. So as I'm discussing relative free energy perturbation calculations in this particular talk, we know these are highly accurate for structurally related or congeneric ligand series. So this really makes this tool really great for lead optimization, especially as I've just said with using a star graph, because what we can do is look at a parent ligand that we put in the center of the star graph, and we observe that all of the ligands at the edges of the spokes of this wheel are ligands that have R group differences. And these are the ones that we want to explore to see if we can actually find anything that will give us a really good high affinity binder. So this method using the star graph is really suitable for triaging a large number of compounds in Flare FEP. Excuse me. And I just want to emphasize that in Flare FEP, we can go up to as many as 500 compounds in a star graph like this. Okay, so after the star graph mode, I'm then going to move on to a normal production mode, which is a multi-connected network. And this, as I just showed, contains more links, and this gives us greater error analysis, basically, because we have more cycles and more redundancy of links. And what I'll do then is take forward the highest affinity binders for a multi-connected graph. As these are going to be input from my Spark initial experiment, they're already aligned. as you can see here, just showing just 10 of the Spark results that are overlaid really closely. If I show what I see in Flare FEP, just overlaying 200 of my Spark results, you can see that they overlay really closely, except in the R group region where there's quite a lot of variance. So this is actually really useful to go from Spark to FEP in this sense, because we have really nice overlay of our ligand data set, which really helps with the atom mapping and setting up the perturbation graph, which is in the initial stages of our FEP calculation. So that's the workflow. I'm just going to dive in now to the background. Unfortunately, I'm going to look at a target that unfortunately needs no introduction. This is SARS-CoV-2 or COVID-19. And the target we'll look at is the chymotrypsin-like cysteine or main protease, otherwise known as 3CL-Pro or M-Pro, which catalyzes the proteolysis of polyproteins, which is required for the viral replication. So inhibiting this activity impedes the production of viral progeny. So that makes it a really desirable target for developing inhibitors. And also there are high sequence homologies in coronaviruses. So it makes it a really appealing target for small molecule drug design. What I'm going to do here is I'm going to show this case study, which references this JMedChem paper by Hoon Han et al., where a structure-based optimization campaign was initiated against SARS-CoV-2 main protease 3CL-Pro. They demonstrate a process of drug discovery based on a starting point ML300 in PDB7-LME. And what they do is they investigate 41 compounds, providing IC50s, which gives us some measured data for our reference. It's very useful. And we will also start with compound ML300 and PDB7-LME and compare our findings with this paper, where their main discovery is the compound 41 with nanomolar activity, 68 nanomolar activity, in fact. So in fact, one of the first points of investigation in this JMedChem paper, I'm showing here one of their first tables, table 1. And they discover compound 21 here, which is an even better binder than compound 41. It's 63 nanomolar. And it shares really interesting SARs with all the 16 compounds in this table. And I'll refer back to these a little bit as we go through this talk. So we're basically here simulating a drug discovery, which is going to also be based on this first PDB, this 7-LME that contains the ML300, the ligand that is co-crystallized here. So this was also the paper author's starting point, ML300 in the box, or in other words, call it compound 1. And this was originally developed by the paper's authors in the aftermath of SARS-CoV-1 outbreak in the pursuit to develop a non-peptidic, non-covalent inhibitor for SARS. So as there's a 96 sequence identity between SARS-CoV-1 and SARS-CoV-2, 3CL-Pro, then similar specificity was assumed, and ML300 here, compound 1, was deemed a good starting point for developing designs for COVID inhibitors. So as such, this table explores amide variants and heterocyclic modifications based on the R group modifications here of ML300, or in other words, compound 1. Right, so for any computational experiment, one thing that's integral to get really good results is to prepare your system. So I'm going to talk about that a little bit. So in solution and in biofunction, 3CL-Pro is a dimer and it's catalytically active. So what I'm showing here is our starting point 7-LME colored by B factor or temperature factor, consisting of 2 proteomers. And here is the side view, which is then flipped over to look at the top. And here it is colored by blue for chain A and pink for chain B. And I just want to emphasize that there are beta barrels that contain the active site in the 2 lobes on this left-hand version. And then we look at the top view, we can see domain 3, which shows off the dimerization, which requires alpha helical domains, which really enable this dimerization. So what I'm showing here is a little bit focusing in on one of the binding sites, so chain A. And we're going to examine more closely the binding site residues colored by B factor. So going anti-clockwise, we can see that there's a high B factor on the ML300 ligand, exactly where we'll be making our R group modifications. So there's possibly high mobility here. And similarly on some of the amino acids that are interacting with ML300, in particular, a serine 46. We see quite high B factor. In contrast, on the other side of the binding site, we have quite a lot of blue B factor or low mobility. And one thing I want to point out is that there is a low B factor here on serine 1, which is actually in chain B. If I superimpose chain A is in blue and chain B is in pink, we can see that serine 1 from chain B actually interacts with some of the amino acids in chain A. And it's basically just really important that for all our computational modeling going forward, we look at the dimer. So after preparing the protein, and that is adding hydrogens, checking protonation or tautomeric states, and or tautomeric states, capping and optimizing, what we're going to do now is examine some key binding site interactions. So going from clockwise, starting with the catalytic cysteine, this is highlighted as it's important in catalysis. We also have a histidine, which H bonds with the benzotriazole on the ML300. We have the serine on chain B, which we just looked at, interacts with some amino acids on chain A. And we also have a glutamine 166, which hydrogen bonds to a carbonyl on the ML300. And I just want to highlight this because this is a really highly conserved residue actually in all coronaviruses. There's also a glutamate, which moves to sandwich the thiophene ring in the ML300 between two methionones. And we also have serine 46, which we just looked at, which hydrogen bonds to the cyclopropyl annelida amide moiety, which is, again, where we're going to make our R group modifications. And we have histidine that makes an edge-to-phase pi interaction with the RR ring on the ML300. So just a bit of background, there is thought to be a catalytic dyad between cysteine 145, which is a nucleophile, and histidine 41, where the covalent binders interact with the catalytic cysteine 145, and the histidine deprotonates the thiol side chain of the cysteine so that it can then make its nucleophilic attack on the peptide substrate. So that's the endogenous process, but we want to be able to inhibit it. So obviously these residues are really important, and we have to give them careful consideration when we're preparing them for calculations. And one particular example I would like to focus in on is it actually really matters where you put the H on your histidine of His41. So if we compare PrEP 1, where the proton is on the delta position of the nitrogen versus epsilon position of the nitrogen, then at first sight, PrEP 1 shows more protein ligand interactions versus PrEP 2. However, if we look into this a bit further and we test out the binding modes by running molecular dynamics for the different PrEP 1 versus PrEP 2, then even though there are less histidine interactions for PrEP 2, if we just pull out the RMSD of the ligand, which is what's shown in this graph here, it oscillates at on average about 1.5 angstroms. Versus in PrEP 2, where it creeps up to about 3 or 4 angstroms. So basically what we're seeing here is in PrEP 2, the ligand is less stable, it's less happy, and it's moving further away from its binding mode. Another way to look at this is to look at the molecular dynamics and examine the protein ligand contacts, which is something that I've done here, where the tables basically sorted by the percentage of friends present. So the interactions that are listed are here given with the percentage of times that they appear in the snapshots of the MD simulation. And what we see is that the glutamine that is highly conserved is strong presence in both PrEP 1 and PrEP 2. But also in PrEP 1, you can see all of these are all histidine 41 interactions, much more prolific in PrEP 1 versus PrEP 2. But then in PrEP 2, we see a serine 46, which, if you remember, we were just looking at the B factor, which had potentially high mobility. And the serine 46 actually binds at the region where we want to make R group modifications. And we see in PrEP2 that with this preparation of states, we actually get better interactions at this region, which is essentially kind of what we need to keep the ligand in the binding mode, to keep the ligand binding nicely, to have a high affinity. So that's all really helpful information. But if we need even further confidence in choosing the right prepared state for our binding site, then another thing we can do is actually use FLAIR to plot the electrostatic complementarity, or EC, of the ligand to our different prepared protein states. So here again, I'm comparing PrEP1 with PrEP2. So we see the different prepared histidines. And what I've got here is a mapping of the EC, where essentially the green shows complementarity, white is neutral, and red shows an electrostatic clash. And you can see in PrEP1, we have quite a bit of red around the histidine. And if I just flip those over, so we're looking down on the different histidines, then you can see in PrEP1, there's quite a bit more clash and a bit more red, which follows where the histidine actually lies. Versus in PrEP2, where you just get this really nice path of green, which tells us this whole region is complementary in terms of electrostatics. So that gives us further confidence that PrEP2 is our correct state for this biological problem. So now that everything's prepared, we can just dive into our computational experiments. And I'm going to do that starting with Spark. So in our Spark experiment, we're doing R group exploration, and we're simply going to load in our PDB code 7LME, and we're going to select in our ligand, ML300, as the reference, delete crystal waters, and then simply select the region, the atoms that we need to discard for R group replacement. So for our R group modifications, we know that the cyclopropyl moiety binds well from our PDB reference from our examinations. But what we want to do is use Spark to examine if we can actually find something that performs as well or even better than the cyclopropyl moiety. So we're going to choose to look for ring atoms only and carbons only in order to find some alternative. And we're going to look through Chembl and commercial databases, which allows us to search around 100,000 fragments. And we can actually do this just in minutes. So the results that we get from this are essentially, we get initially 1,000 hits. Some from the paper are discovered, so I'm just referring back to the table again. So compound 8, 15, 19, 20, and 21 were all discovered in the first 1,000 hits from Spark. But what we did next was take forward just the highest 250 as sorted by BIF, where BIF represents the percentage of similarity to the starter molecule. Now, there are lots of ways to sort and filter according to clustering or molecular weight or docking score perhaps, or even EC, so electrostatic complementarity, which we just looked at. You can create scores and then sort and filter according to those scores. But this is a very simple, very quick experiment, and we're going to keep it just really straightforward by only taking forward the top hits of alternative rings based on the field similarity to our reference, ML300, or in other words, compound 1 in the box. So now we're just going to move on to the FLAIR FEP, or in other words, free energy perturbation experiments, in order to accurately predict binding free energy. So we're taking forward the 250 compounds discovered by Spark, as I just said, sorted by the BIF score, but also with a charge that is 0. What we need to do for a star graph is to choose a center of the star, which we will have as compound 9, which is, luckily for us, a molecule with known activity that we've taken from the table I've been referring to. And it's got a very fortuitous R group that is just a hydrogen. So it's particularly relevant to be the parent molecule in our star. If we make a star graph even with no intermediates, like this one on the right, you can see quite a densely packed parent to ligand direct linking. Versus if we use automatic intermediate generation, we get intermediates generated that allow us to make some of the more complicated transformations more easily. So within Flare FEP, we have this ability to let the software just do this automatically without any user interference. So we're going to take advantage of that. And that's what we did here. We got 250 ligands from the Spark experiment. We generated intermediates automatically, just clicking a button. And this gives us 400 ligands in total. So there's an awful lot of ligands we're going to test. So we're going to do quite a short 1 nanosecond simulation and just use the single way mode that I discussed. Of course, we also have to use our very carefully prepared protein state in order to get decent results. So that was the first stage. We then filtered out some of the problematic links. There were a few that contained fused rings and ring breaking, which is just a little bit too tricky for some of our transformations. So then we selected the top 200 with the highest affinity. Again, added intermediates to our star graph. So we got 300 in total. And again, did a 1 nanosecond single way simulation. From this, then we took forward the top 40 with the highest affinity for a normal multi-connected production run. So ML300, in other words, compound 1 and compound 9, were actually both included this time as known actives in generating our multi-connected perturbation graph. And the reason for this is kind of to simulate more of a real life experiment as much as possible, where hopefully you're going to know at least 1 to 2 binders. The more the better, usually. But often you might not have that much information. You might just know 1 or 2 known actives. So this is the kind of situation we're simulating. In the end, ML300 or compound 1 was not actually used in the graph, as compound 9 actually links the best to the 40 ligands that we have. And we get 43 in total, including this compound 9 in blue and the 2 intermediates that we generated using, again, the automatic intermediate generation. This time we're going to simulate for longer using 4 nanosecond lambda windows, and we're going to do now a dual way calculation. So then, taking forward the most promising 16 compounds from this, we did what I'm going to call a refinement of the network, and this can include adding links to the cycles, perhaps adding even more intermediates, and also increasing lambda windows where required to smooth out any poor overlap. And what I really want to emphasize is that we actually discover compound 21, which, if you remember, is the highest affinity binder from table in our paper reference. This is quite exciting. It's the highest measured binder in the paper table, but it's also the strongest predicted binder with the highest affinity directly from our initial 100,000 fragment wide search. So that's the main result, but just to go into a little bit of detail, further reduction of the compounds in the graph and sampling for longer, so going up to 10 nanoseconds. And if I circle this group of ligands up in the FEP graph here, these contain cyclopropyl moieties that are similar to ML300, but are rearranged slightly. However, if we compare to ML300 with minus 7.2 kcal per mole, they have higher predicted affinities of minus 8.2 kcal per mole, which gives us possible other avenues of research, which is exciting. Further, so compound 21 is the main discovery. It's this highest binder. But what we also observe is actually potentially an interesting activity cliff example. So we have compound 21, which is at the top end of the binding. But we also have another compound, which I'm going to call compound 20-like, which is at the more of the lower end of affinity. So we have minus 8.9 kcal per mole versus minus 6.7 kcal per mole. And this is largely to do with the seemingly innocent methyl group addition that we have here. So just to go into this a little bit more depth, and referring again back to the paper table, there is a set of heterocyclic azole nitrogens which demonstrate this type of activity cliff. So we have our highest affinity binder, compound 21, and then methylating the NH, we actually get compound 20. And you can see this reduces the binding by 3 kcal per mole, which is quite significant. And similarly, if you compare 19 and 18, again, the methyl group completely diminishes binding. And this is all observed by the paper authors, but it's interesting that we see a similar effect in the FEP calculation. If we compare compound 21, which we picked out as the highest predicted binder. And then we compare with compound 20-like. I'm calling it 20-like. It's not quite the same as compound 20. There's a difference in the placements of the nitrogen and methyl, but it demonstrates a similar principle and has unfortunately placed methyl group. So the point is a very small, seemingly small structural change can convert the best binder to a much less good binder. And this is reflected in the FEP results. It can largely be explained, as noted by the authors, actually by a loss in a cysteine and threonine interaction and a steric clash, and consequently in moving out the binding mode. But as relative FEP calculations assume the same binding mode with the excuse me, with the larger sampling calculations, this difference in binding is actually reflected in the longer sampling and captured in the FEP results. So it's nice to see that there. I'm going to round up now. I think I'm getting to time. The best binder, compound 21, was efficiently and successfully discovered using this workflow. So that's our main result, but also what's exciting is that other potential active compounds are found, opening up alternative avenues for development, such as the ML300 light molecules with the repositioned cyclopropyl group. Also, we only concentrated on R group modification in this area, but we could then fix our NH result for compound 21 and then move on to look for replacements of the thiophene ring, for example, which is indeed similar to what the paper reference does in its discovering of compound 41. We could also look for something that specifically binds the reactive cysteine 145. This is not something that's been immediately taken advantage of, at least not in this study. So there's lots of interesting options that open up. Hopefully we've also shown that flare FEP can excuse me, reliably, efficiently, effectively post-process results from Spark. And we're showing a workflow that really streamlines drug discovery. It explores novel ideas, correctly identifies the most promising compounds, such as compound 21. Excitingly, this is all done starting with very little information. So we just needed 2 known actives that we put into our FEP graphs and 1 PDB crystal structure, which we started with. And that is it. I just want to say thank you to Stuart, Nathan, Mark, and Giovanna. This was all teamwork done for this project. And please get in touch if you have any questions or queries, but very happy to take questions now also if there are any. Thank you.
All right, thank you so much. I think there's one question in the chat from Victor. For the sake of time, I'm going to just read it out. How would one account for the situation where HIS41 protomer changes with different ligands? Should one run both HIS41 forms, or how would you account for the changing of the reference protein state in relative free energy calculations?
Sorry, could you just repeat that for me again?
Sure. Actually, here, why don't I unmute Victor?
Okay.
Victor, I'm going to unmute you if you're still, oh, that looks like.
Yep, yeah. Can you guys hear me?
All right. Hey, Victor, thank you.
Hey, Jennifer, thanks for the nice sort of preview of how you guys implemented the FEP in Cresset. It was a good example, I think, as well, about some of the subtleties of how the protein state is set up to start the calculations, as you explained. But my question was related to, we showed the specific protomer of histidine being the good one, for one of the ligands. But what if that changes with another ligand, right? So if you bind a different ligand,
yeah.
How would you account for now different reference state now? Like, okay, we can probably do the simulation for each of these ligands and see which one is more stable. But then we're changing the protein reference state in FEP calculations, right? So what would one need to do with that?
That's a very interesting question. So I think for this particular example and examples like this, it's possibly more important to have a longer sampling time than the default. Generally in flare FEP, we use a default of 4 nanoseconds for the lambda windows. But if you have a situation where a flipping of a histidine, or even the tautomeric state of a histidine is up for question, then if you can sample timescales where the flipping would happen, with respect to each ligand that you're having transformed in your calculation, then that would be for the best. So I would recommend the best thing you can probably do is to do sampling for longer to increase your lambda window length. But apart from that, if you have a really ambiguous protonation state or tautomeric state, that very much depends on your ligands set, then that can be a real challenge for FEP in general. So that is a tricky situation and just something that you have to consider when you look at your ligand data set and you prepare your protein.
Yep. Okay, thank you.
Thank you.
Are there any other questions? If not, let's all thank our speakers one more time. This is a really, really good session. Thank you guys so much for both your talks. And yes, please feel free to reach out to the emails that they sent. And if anybody else would like to be in contact with either the speakers or the BAGIM group, you know where to find us. Find us on the meetup group. Thank you guys so much, everybody, for coming. Have a good one and enjoy the rest of your day.